Welcome back to the playlist on nucleic acid catabolism. And in the last video, what we did is we looked at the function of this enzyme that was called urate oxidase. Okay, so we looked at the function of urate oxidase. And there were several things that were important about this enzyme, but the most important one is that it's not expressed in humans and upper level primates and hominids and things like that. And if you want more review about all those things related to urate oxidase, I'm doing a quick recap now, but certainly go back and watch the previous video. So the idea behind urate oxidase is that humans have the gene. We have the gene for urate oxidase, but keep in mind that genes lead to the production of a protein, or in this case, it's an enzyme. But humans do not express that. So that means that the gene for urate oxidase in humans and upper-level primates, great apes and so forth, is transcriptionally inactive. So that's important. It's transcriptionally inactive. And all that means is that RNA polymerase does not transcribe the gene. So you don't get mRNA, and therefore if there's no mRNA, there's no protein. So that's the big idea. Humans do not make urate oxidase, which basically implies that the end product of purine catabolism and purine degradation is urate. This guy right here, very, very important. Okay? And again, if you want more details on urate oxidase, certainly go back and watch the previous video. But what we're going to do in this video is we're going to go over the physiology, the organic chemistry, and the biochemistry of urate. And what you may see it referred to as most often is uric acid. Okay? And there's sort of a... Usually you'll see these words used interchangeably, urate and uric acid, but there actually is a physiological difference between the two. And the difference lies in the fact that uric acid literally is a weak acid. So what do we mean by weak acid? Well, we simply mean that the deprotonation of uric acid does not go to completion, meaning the equilibrium lies very far in the direction of the acid. So if you have the acid form, so this is the conjugate acid form, and you have the conjugate base form, if it's a weak acid, that implies that the equilibrium lies pretty far in the direction of the acid. And in fact, in most cases, you'll find uric acid in the acid form. But what I want to do is think a little bit about which one of these protons on uric acid is the one that's most ionizable. In other words, which one has the lowest pKa? Well, if I look at this molecule, notice that it has several protons on it that are good candidates. You have this one right here, you have this one, you have this one, and then you have this one over here. And the question I want to think about is which one of those possibly is going to have the lowest pKa. Okay? And to do that, you sort of have to think about, if it was deprotonated, where can I distribute the negative charge among the most number of atoms? Okay? So uric acid in this form, this is the uric acid form, it's not very soluble. Okay, so that's important to realize. This is what we call non-soluble. Okay, this is not a very soluble molecule, but it becomes more soluble when you ionize it. So the question is, which proton does a water molecule attack in the blood? And the answer is that it's going to, I'll do the mechanistic steps in green, it attacks this one. Okay, and so you have to imagine that, basically this lone pair can kick in right here, and you can force the electrons onto that oxygen atom. So that's one direction that those electrons can go. So basically what you're doing in that case is you're distributing the negative charge about this system right here. But I'll do another electron movement in red just to show you something, that the electrons could also move this direction. So you could kick the pi electrons in right there. You could kick these in right here, and then you could kick them onto the oxygen atom. Okay? So whenever you deprotonate this particular nitrogen right here, those electrons can go in multiple directions. And actually what you'll find is that when you deprotonate that nitrogen, you get this right here, which is the resonance hybrid. Okay, let me do this in orange. So the negative charge in the electron system is distributed about all this right here. Okay, so one of the keys about determining pKa's is if you can somehow create some kind of conjugated pi system, okay, and you can move those electrons basically around the system, you've created a more stable anion. Okay, so the negative charge, notice, can be distributed over this oxygen, it can be put on this nitrogen, it can be put on this carbon, and it can be put on this oxygen. So because I can distribute the negative charge over 4 atoms, that makes this particular conjugate base, or the anion, more stable. Okay? Now, it won't exist all in this form, right? Because the uric acid was a weak acid. Right? This is a weak acid, but some of it will exist in the conjugate base form, and this one is the most stable conjugate base, and specifically this is where the term urate comes from. This is where the term urate comes from. This is more specifically the deprotonated form of uric acid. Okay, but I want to make it perfectly clear that most of it's going to exist as uric acid, not urate. Okay, so there is a difference between the two technically, but most often when we talk about these molecules, they're equilibriums between each other, so we use the terms interchangeably. Okay, now what I talked about in the last video was that what can happen essentially is, let's say you're eating a high purine diet. Okay, I mentioned that in normal cases, okay, you eat excess purines, and some of it might get incorporated into DNA, others might get incorporated into RNA, and the rest that you don't want floating around goes into the catabolic pathway, as shown right here. Okay. And you'll end up generating some urate, right? And then it doesn't stay in the cells that degrade it. It goes into the blood. So what you'll end up with initially is something called hyperuricemia. Hyperuricemia. And all that means, it's a fancy word that just means elevated uric acid in the blood. Okay, so this is an important term, elevated uric acid in the blood. And I mentioned that in a healthy individual, there were some proteins that existed in the nephron that allowed tubular secretion, basically, of uric acid, so you could get rid of a lot of it. And there's also proteins that could basically allow reabsorption of uric acid. One of those that allows tubular reabsorption of uric acid is called SLC2A9. Okay, and actually there's a series of proteins here that exist in the membrane of the nephron. Okay, and the ones that are shown not in this sort of bold color right here, these are the ones that basically are known to allow tubular reabsorption. Okay, so for example, as I mentioned earlier, the SLC2A9, this basically allows tubular reabsorption of not only glucose and fructose, but also uric acid. And there's two types of it. There's a V1 that exists on the basolateral side, and there's a V2 that exists on the apical side of the nephron cells. Okay. There's also some other ones here and there, like for example, there's a Urate 1. Okay, that's theorized more or less to, the direction is more or less unknown, but it's thought more to act in the reabsorption direction. Okay, there's also two proteins right here that allow tubular secretion of uric acid, so secretion into the lumen of the nephron, and then that gets excreted in urine. Okay, but basically the idea behind all these proteins is, if you have a buildup of uric acid, it's likely due not just to the fact that you have excess purines in the diet, because as we know, most people have lots of purines in the diet and they don't get gout, right? So it likely has to do with something over here. some dysfunction of some of these proteins. And actually what it's actually known that people that eat lots of this guy right here, which is high fructose corn syrup. People that eat lots of high fructose corn syrup are more likely to develop gout. Well, that begs the question, what is gout? All gout is it's a buildup of crystalline forms of uric acid. We'll go into the details of gout in just a few minutes, but I think the idea to hold in your mind is gout is basically a crystallization. It's a crystallization of the molecules of uric acid. Okay? So it's known that people that eat lots of high fructose corn syrup, they tend to get gout. And here's just sort of a theory as to why that happens. Notice that the SLC2A9 functions in uric acid reabsorption. Okay? So the idea is that, let's say you have a lot of fructose here. Okay, fructose is also known to be reabsorbed by SLC2A9. Okay, well maybe perhaps if the cell senses that there's a lot of fructose, perhaps it upregulates this protein. It's just changes in genetic expression of this protein. It upregulates it, and so if it upregulates it, it also reabsorbs uric acid. And so more uric acid gets reabsorbed, and therefore more gets into the blood. Okay. Also, another really important thing that we'll go into more detail in just a few minutes is that people who have a higher acidity in the blood, their kidney gets affected. Okay, so high acidity in the blood, okay, is correlated with kidney dysfunction. So basically, if you have high acidity in the blood, what ends up happening is you have problems excreting this guy, which is ammonium. So people that have high acidity have trouble excreting ammonium. So what ends up happening is you get high ammonium in the blood. High ammonium in the kidney and high ammonium in the blood. Okay? And this acidity that's present because you have trouble excreting ammonium leads to uric acid in this form right here. And what did we say about it? We said it's basically not very soluble. Okay? And so in this form, whenever you have excess uric acid in the blood and it's not in the soluble form, it tends to crystallize. Okay? It tends to crystallize. We'll go in more detail on that in just a few minutes. But I just wanted to kind of have you bear in mind that when you do have excess uric acid in the blood, it tends to form polymers with itself. Okay? And that's what results in the crystallization process. And it will actually come out of solution and sink to the lowest possible point in the body. Okay, but high fructose corn syrup impairs uric acid secretion into the lumen of the nephron. High acidity in the diet tends to inhibit ammonium secretion, and so the acidity in the kidney goes up, and uric acid stays in the non-soluble form, and it impairs its secretion into the kidney. Okay? So now that that's out of the way, let's actually look at the crystallization process. So one of the things is that uric acid and compounds very similar to it can actually form dimers and tetramers with each other. Okay, so this right here, this is not exactly uric acid, okay, but at least it should give you an idea of how these purines can sort of conjugate to each other. Notice right here that this particular oxygen has a partial negative charge. Okay, and so does this one. This has a partial that should be a negative. This has a partial negative charge as well. And then notice also that this hydrogen has a partial positive, and this one also has a partial positive. So notice what's happening here is you're getting hydrogen bonding between certain points in different urate type of molecules. Okay, and this happens with all sorts of purine type of molecules. It happens with xanthines, as shown right here. Okay, these are methylated forms of xanthines, but the idea is that urate-like molecules can hydrogen bond with one another and form really stable interactions as dimers. And here's another picture. They can also do it as tetramers. These are basically, these are similar to uric acids. So there are 4 different arrangements that you can have, but notice in each case that you're getting hydrogen bonding between adjacent uric acid molecules, okay? And we can, of course, debate on which one's the most stable, and to be perfectly honest, probably all of them exist to some extent, but these tetramers and these dimers of uric acid, these all represent the basic monomers that make up the polymers of uric acid crystals. And that leads us to, what do the crystals look like? Well, these right here, these are the uric acid crystals. And notice how they have literally come out of solution. So that's the thing about the crystallization process, is literally the crystals have come out of solution. Now here's the thing about the uric acid crystals. Number one, they are more dense than water. Okay, so in other words, their mass per unit volume is greater than that of water, and so as a result of that, they sink. They sink in the blood, okay? And in general, they sink to the lowest possible point in the body, and that guy happens to be, usually in most cases, the big toe. Okay, so in most cases when you hear somebody with gout, it's usually in the lower extremity. Sometimes it can be in the knee, but in most cases you'll hear this in the big toe. And let me circle it in red because it's bad. And the reason it's bad is because it's excruciating. It's one of the most painful diseases that you can have. Okay, so whenever you have excess purines that have trouble getting excreted, it crystallizes in these dimers and tetramers that were shown over here, okay? And when they come out of solution, they polymerize and they sink to the lowest possible points in the body. And part of the reason they're able to crystallize is because they're not very soluble in water, especially in the protonated form, which is what we had shown up here. This is the protonated form, not very soluble at all, okay? And if that wasn't enough, you can actually get crystallization processes that occur in the kidneys. Okay, so what you can actually have are things called calcium oxalate salts. Let me write that down. So you can get things called calcium oxalate salts. Calcium oxalate salts. And actually what can happen is you can actually get calcium oxalate salts due to seed crystals of urate crystals. Okay, and basically what happens is the seed crystals of urate, okay, actually facilitate the crystallization process of calcium oxalate. Now what I'm going to do is I'm going to do something I normally don't do in these videos. I'm actually going to show you a pretty good video on what it actually looks like in a very accelerated time of how seed crystals actually function. So this is what I'm about to show you. This is a video of seed crystals, so I'll try to shut up and show you exactly what's happening here in the kidneys.
This is our solution of sodium acetate, and I have it covered here because even the tiniest little speck of dust will trip this. So watch this. I'm going to take it off, and I want you to reach out and just get one tiny little crystal, just a single one. Show on your finger. Just a single little crystal. That's like just a couple of times. That's it. And just reach over and get it. Just tap it right on the top, right? So just tap it on the top there.
It's just sprouting out, kind of like when you say things freeze.
Absolutely. And so you've got crystal formation in there, and it's called sodium acetate trichoride. It's also exothermic, so put your hands around it and get nice and warm.
So that right there, that's basically the process of seed crystals causing the formation of other crystals. And so the uric acid crystals can actually cause crystallizations of these calcium oxalate salts. Okay, and calcium oxalate salts actually are the basic foundations of kidney stones. These are the foundations of kidney stones. And kidney stones are some of the most painful things that people have when they end up getting passed. And so a lot of times you have to have things called lithotripsies. And what lithotripsies do is they basically break up the one big kidney stone, they break it up into smaller, more easily passable stones. But they're still painful. And some people, I don't know this because I'm a guy, but some people actually compare passing a kidney stone to giving birth in terms of the pain that it causes. And we can, debate on that, whether or not it's as painful. But the idea is that seed crystals of urate can actually cause the crystallization of calcium oxalate salts into kidney stones. So let me circle that in red. So these kidney stones that form are extremely painful, and they can be induced by uric acid. Okay? So hopefully this video gave you a little bit of intuition on uric acid. One of the other things that I'll mention about uric acid that it can do is it can actually act as a free radical scavenger. That's actually going to be the topic of the next video. Okay, one thing that uric acid can do, and this is actually what is thought to be the benefit to humans and upper level primates and hominids and so forth of not having urate oxidase. So remember, keep in mind always that humans do not have urate oxidase. Okay, but the benefit of not having that is that you do have higher levels of urate in the blood than, say, other animals. Okay, so other animals are going to have significantly lower levels of urate. And what we're going to talk about in the next video, which is part 3 of our uric acid and urate talks, is we're going to talk about how uric acid can actually scavenge for free radicals. So hopefully you join us for the next video, and I hope this video gave you a little bit of intuition on urate and urate oxidase. See you in the next video.